Hello and welcome to Principle of Cognitive Neuroscience, Part A. As you may know, the course is divided in three parts. Main models, where I will explain how people imagine how the brain works, whether this is a brain that is fractionated in separate regions dedicated to different functions, whether those regions need to organize themselves in theory, one after the other, in a hierarchical organization to achieve the function as we know it. Is there something more through this exchange? Is the association between brain regions more than the addition that the activity? So this will suggest an integration level through this interaction, and this is all that is behind the holistic vision of the functioning of the human brain. And today we'll mostly speak about glial models, which are a little bit of the forgotten cells in the brain. We'll speak also about epistemology in the next lecture. The second part of the course is dedicated to main concepts, where we will tackle what constitutes a result. When do you stop and you're like, okay, I got a main result about the brain that I can publish. So we'll give those examples. And then not recorded, unfortunately, and you have to assist those courses. Will be the practicals, where I'll teach you how to manipulate informations from the brain, whether this is neuroimaging or review or just data. I'll explain to you how you can do the job itself and produce results. Without further ado, we're going to start with the glial model up right here. And as you may know, and we discussed that before, the structure of the brain is made of neurons and connections. And those neurons activate themselves and convey information through their axons to other neurons that can be close, distant. They can be close, like very close, like next to each other, or they can be in a different gyrus. So you get a skip a sulcus, as you can see here in orange, or you can directly go into a different lobe of the brain, from the frontal to the parietal lobe, for example, or from the frontal lobe to subcortical structures. Those connections can also go to the opposite hemisphere, and together these exchanges define the functioning of the human brain as we know it. Or do they? Or did we forget some key players in this exchange that happen in the brain? Well, apparently research indicates that we might have forgotten a little bit or put aside some very important actors in the functioning of the brain by being allowed to focus onto this neurocentric vision of the brain. So as you may see here in talk, you have those neurons that are interconnected between each other thanks to synapses that will transmit neurotransmitter from the end of the axon to the beginning of the new neuron, so it's dendrites. But this communication and the activity of neuron are not really isolated and require supporting cells that are doing a lot for the functioning of the human brain. For example, the neuron, in order to produce its action potential, will need some nutriment that can only be provided by the astrocytes that you see at the bottom of this slice in blue. And in order to convey its information along its axon to other neurons, you will need oligodendrocytes that will myelinate the axon in order to speed up its conduction. Really, the key behind those cells, which are called glial cells, have been discovered by fascinating women in science. And instead of really probing like her idea and the way she demonstrated, I thought it would be a good idea to show you a movie about life and achievements, and hopefully you'll find inspiring, and you will learn from her and live with us through this movie how we discovered the importance of glial cell for the functioning of the human brain and the very definition of our individual differences.
It is the most magnificent structure on this earth. A full human brain that you can hold in one hand. Nothing can compare with it. Those cells can produce ideas. Have you ever thought how a cell could produce an idea? This is who you really are. Take away the brain, you take away the person. I've spent more than 60 years studying the brain. And it was pure joy. Don't you just love the brain?
Marion Diamond's lectures on YouTube have over 1.7 million views, making her the second most popular college professor in the world.
Before Marion Diamond's research, the accepted scientific understanding of the brain was profoundly mistaken on a key point.
Joining us now, Dr. Marion Diamond. She has spent 30 years studying the brain.
Konnichiwa, Marion Diamond.
Marion Diamond is the author of the first study ever published about the brain of Albert Einstein.
It is my pleasure to introduce Professor Marion Diamond.
So welcome to our great university at Berkeley. How many of you have studied brains? How many have studied human brains? How many have seen a human brain? For those who haven't seen a brain, well, when you see a lady with a hat box, you don't know what she's carrying, do you? And there you are. Did you know that this is what you hope you look like inside? The major parts, you see the cerebrum, These deal with higher cognitive processing, for you to be thinking about what you want to do this evening. It may be coming out from your frontal lobe. Then you have a little brain, a cerebellum. How many of you have heard about a cerebellum?
Things are just flooding onto my brain right now to share. The marvelous things that this brain can do. This 3-pound mass that sits in there. Everybody wants a better brain. There's no doubt. If there's an opportunity to improve your brain, you want to be there. And so, 5 items that we have found are essential for a better brain. I think of all the millions of things, but 5 shown in our work. And the first was diet. The second was exercise. The third was challenge. And the fourth was newness. And the fifth, we've added love.
When somebody comes along with an entirely new idea that shatters the old one, this shatters a very important part of us, part of our internal world. And by nature of the beast, it's bitterly fought.
Scientific advance doesn't occur on a linear continuum. It occurs in fits and stops and jolts and cliffs and abrupt changes. And I think Marion's work was a classic example of that. I think most people would have said, why are you doing that dumb experiment? You're just wasting a lot of rats and a lot of rat food. Fast forward, guess what? That work has really spawned an entire generation of research, helped initiate this whole field.
Marion Diamond is one of the founders of modern neuroscience. Over the course of her career, concepts which we now take for granted, such as brain plasticity and enrichment, were controversial scientific battlegrounds, where she and her team challenged the accepted theories about the brain's potential. Back in the 1960s, the scientific consensus was that genetics determine how your brain turns out, that your brain's nature is something you inherit. Genetic determinism, which viewed the brain as static, fixed. The brain's maximum potential, determined at birth, and unchangeable.
Well, certainly all the experts at the field until that time had taught and taught us that the brain followed a genetically programmed pathway of development, and that when it reached the point where it was programmed to reach, that was it. And then the next step was a gradual decline as one got older. And Marion's work went so against the grain, it was totally anti what we had all learned to accept.
At age 31, Dr. Diamond was a part-time researcher, part-time lecturer, and mother of 3 with another on the way. She joined a team of 3 University of California, Berkeley researchers who were looking for evidence that the brain is affected by one's environment and not predetermined only by genetics. They would work together for the next 15 years.
Kresch and Rosenzweig were psychologists, and Bennett was the chemist. Then I filled out the team by becoming the anatomist.
Ideally, I would like to be working with human brains. I have yet to find anyone who's willing to give me their brain to work with. Is this audience any different from the others that I talk with? Therefore, we go to a smaller animal, the rat.
The studies were simple, yet they were elegant, like many great experiments. She simply had animals in a enriched and a non-enriched environment. In an enriched environment meant in their cages they had more toys and balls and wheels to play with.
to raise rats in these, what she called, enriched environments, which I think of as the Disney World of rat environments with lots of toys and lots of areas to run around, and simply ask, what does that do to the brain's anatomy?
We have what we call an enriched environment. It may not seem enriched to you, but for rats, we have 12 rats living together in a large cage, and they have novelty stimulus objects to interact with. They each have a brother in a standard colony. Or the impoverished condition, no toys and no other rats.
A simple investigation. 3 environments, 3 sets of brains, then after 80 days, compare. If genetics determines brains, they would all be the same size. But if there is any measurable anatomical difference, that would decisively demonstrate the importance of the environment.
But I didn't know, how was I going to measure the anatomy? How to do it? I tried cutting out a little piece of the prime area of the brain. That didn't suit the bill. So I just sliced the whole brain across. Then you could pick up the outer layers of the cerebral cortex. And just measure them under the microscope. Sure, it takes months and months to do this. People said when I started, well, that'll take you 2 years to get a good answer. I said, I don't care, as long as we get the answer. It all took time. Our most important finding showed that with enrichment, the brain increased dimensions, and with impoverishment, it decreased. A 6% difference. It was thrilling. I ran across campus to show Dr. Kresh. You don't see me normally running across campus, but this is something that had never been on this earth before. He looked at me and said, "This will change science." Yes, the brain does change with experience. Nobody else had made such measurements and found these results. We were terribly excited about it.
This scientific breakthrough was remarkable, not because Marion was the only scientist to suggest these new ideas, but because she was the first with evidence, data, actual measurements in a real brain that confirmed the brain is not entirely determined at birth. It changes, responds, reorganizes. The scientific term is plasticity. And in the years since, brain plasticity has emerged as one of the core concepts in modern neuroscience.
Brain plasticity is the idea that the environment, the outside world, can actually change the brain's anatomy, physiology, and function.
What it means is that the brain is not fixed, immutable structure. So for instance, one study was the size of spatial memory areas in the brain become bigger in taxi cab drivers.
Another example. Regions of the cortex associated with playing music are larger in musicians than non-musicians. Short-term plasticity. If you wear a blindfold for 2 days, your visual cortex reorganizes to process sound and touch. With stroke patients, plasticity enables healthy brain cells to take over the functions of damaged brain areas, allowing recovery of lost abilities, such as speech, movement, and perception.
Plasticity actually means increased growth of those nerve cell processes, increased numbers of connections to provide substrate for this more amount of information coming in. So your brain actually structurally changes, functionally changes, information carrying capacity changes.
So there's a remarkable and wonderful potential in the human brain.
And that's the beauty of it.
What plasticity reveals is that we are the masters of our own minds. That each one of us, as individuals, has the potential to change our brains, to become the person we want to be. What we do day to day, minute to minute, grows and builds our brain. What a gift. And what a responsibility, as we literally create our personal masterpiece, our mature brain. When that paper was published, 1964, I spoke to the American Association of Anatomists, the first big conference that I had ever spoken at. There weren't too many women on the agenda. When I finished, a man in the back row, I mean, it was a big auditorium, stood up and shouted, "Young lady, that brain cannot change." So
people didn't think the brain was capable of any degree of plasticity. You know, the brain could change, you know, because you were in some certain environment. You know, that's nonsense.
And my colleague, very brilliant Argentinian, he got red in the face. He says, "This can't happen."
That was a difficult place to be for anybody, let alone a lone woman.
Fortunately, I was taught to repeat your experiments, so I wasn't afraid to say it. I said, "I'm sorry, sir, but we had an initial experiment and a duplication experiment that showed that it could."
In fact, she was right, and the scientific culture at that point was wrong.
Not only she reproduced it, but many other people, and now a whole new kind of generation.
Marion Diamond rang the bell and helped initiate this whole field.
And the interesting thing is that it's taken close to 2 generations for the enrichment paradigm to be really accepted and to be a whole new active area of research. But from my point of view, more importantly, to be sort of noiselessly integrated into the structure of life, a part of our culture.
Enriched environment. From Dr. Diamond's research, to the mass media, to child care facilities across the country, to China, where her ideas have been distributed in pamphlets by the million. It all started here.
And now that we've begun to appreciate the plasticity of our cerebral cortex, we must learn to use this knowledge through enriching the environment for everyone at any age.
So, how? How to create an enriched environment for oneself and one's brain. Dr. Diamond distilled her advice for a better brain into one simple phrase.
With just a little phrase that goes for the brain as well as for muscles. You use it or you lose it.
Use it or lose it.
Dr. Diamond, use it or lose it.
That's essentially what we believe.
With use, you grow branches. With disuse, sitting around doing nothing, you lose it.
The idea is that if you want a healthy brain, you go challenge yourself intellectually, you travel, you exercise, you do all of these things that engage your brain into something, because you either use it or you're going to lose those functions. The brain, as Marion has shown, is plastic and is dynamic, which means do something new. Go find this piece of your brain that you haven't used before and challenge it.
Since her pioneering work, Literally thousands of successful methods of enrichment have been studied and identified by other scientists. Reading aloud to children, running, meditation, bilingualism, playing a musical instrument, staying socially connected, and negative factors that impair the brain. Childhood poverty, fear, trauma, isolation, sleep deprivation, low socioeconomic status, protein deficiency, and of course, if you want the ideal enriched environment, what better place than the classroom?
My name is Marion Diamond, and I've been teaching here for many decades because I love to awaken students to anatomy.
In 2005, UC Berkeley put Dr. Diamond's Intro to Anatomy course on YouTube. 5 years later, thanks to the internet, hers was the second most popular college course in the world.
All right.
1.7 million hits on YouTube.
How many of you know the structural and functional unit of the lung, without which you cannot breathe? One.
Anatomy is hard to learn.
Second question. How many of you know the structural and functional unit of compact bone, without which your bones would collapse? You'd just be a puddle of protoplasm there. How many know the structural and functional unit of compact bone? We do need a class, don't we?
There's just so much vocabulary, so many different items.
Start with the skeletal system. Now we're going to put the muscles on it. Your blood vascular system, lymphatic system, respiratory system, the nervous system, spinal nerves, cerebral, anterior cells, spongy bones, mucous membrane, orbicularis oculi.
And in the worst hands, it could be as boring as reading last year's tax laws.
With subject matter this daunting, what makes this class so popular?
Your cardiac muscle beats about 72 times per minute. Now, I have to do this so that you really know it. We're going to close our fist 72 times. See how your hand feels after 1 minute. 1, 2, 3, come on. 4, 5, 6, 7, 8, 9, 10. You've got fibrillation. 42, 51, 50, 42, 43, 44, 45. You're not doing it back there. 57, 58, 59. This is a hands-on science class. 57, 58, 59, 70, 71, 72. Can you imagine your heart does that for 100 years and never stops?
I can remember that first day of the first class I ever took with her. I saw a science rock star. She made science and learning personal. She made us think about why we were learning. Why do you want to learn about anatomy?
Acetabulum. Say it. I want you to talk, because when you get out of this class, you're going to have to use these terms. You'll say, I see them on the page, but I never used them, right? So I squeeze and squeeze to get you to talk. By the end of the semester, you're talking. Right now, you just sit with your mouths open and sort of look at me. But I know it can be done because we do it every year, and I have to work just as hard. If I have second graders and ask them, they're all talking, they're all shouting. You get inhibited, you're afraid to be wrong. And we're saying it's okay to be wrong. Speak up. What's it called? Thank you.
Dear Professor Diamond, I just wanted to write to thank you for allowing your anatomy teaching sessions to be streamed online. Dear Dr. Diamond, good morning, Mrs. Diamond. Good day, my name is Anita Ferguson. I'm a homeschool mom. I'm a UK expat. I'm a high school student from Pennsylvania, Geneva, and Hong Kong, University of Michigan student, reaching all these people.
1.7 million hits on YouTube. I mean, think about that. People are going out of their way to learn from this woman. It really is special that she can do that.
She always used the blackboard, which I thought was way more engaging. And I still have my notebook that I got from her class.
And I write things on the board because it gives you a chance to think. It's not old-fashioned. It's through years and years of studying education and how you learn. And you learn with kinesthetic sense when you write, so it's in your handwriting. If I just flash a slide up there, you have no time to put it in what we call your association cortex. So by the time you write it out, you can remember it.
Do you really understand how to teach students?
I usually take students to lunch when we have time, and we've got a little time, and the way I can today is I throw out the chalk, and whoever gets the chalk will go to lunch today. And I try not to hit you in the head when I throw it. Do you want to go to lunch today?
Who does that? What professor does that?
Dr. Diamond, so many things I want to talk about. My name is actually a Sanskrit name.
Dear Dr. Diamond, I really enjoy to know more and more about the body, and especially my body.
Our bodies. The subject is ourselves. Us. What we really are. Anatomy reveals and reawakens us.
You ever thought so much about your nose before? Right there in front of you all day long? Look at the human body and what it offers us in knowledge, and constantly changing. I've started it back in, when did I see my first brain? I guess when I was 15. And here I am, 85, and still find it remarkable. And we're teaching and sharing with students. All right, well, I expect you all to get A's now. I've done my job, you've got to do yours. And then remember it, that's will really be fine.
Don't you want to learn about what makes you, you?
We have so many unanswered questions about the brain. What is thought? How are memories made? If you had to choose one brain to study in the entire world, whose brain would you choose? No one doubts the genius of Albert Einstein. But what in his brain enabled him to think so deeply?
I read in Science magazine that Einstein's brain was in a box. I asked him if I could have 4 pieces because we were studying human brains in normal males, and I wanted to compare those areas with his. They said, fine. 3 years, there were my 4 pieces of brain. So we studied them.
In 1984, 29 years after his death, Marion Diamond and her associates were the first ever to publish research on the brain of Albert Einstein. In hindsight, it seems obvious, but no one else had done it. In science, if you want to get answers that matter, you have to find your way to the right question. It's not easy to find a question so powerful that the answer makes a difference.
See, that's something that there's one of in the world. We've got Einstein's brain on my screensaver. How did one feel in looking at Einstein's brain for the first time?
It was a total emotional experience. One of thrill, exhilaration, and yet one of apprehension of what we were really doing, stepping into the unknown of one of the most brilliant minds that's ever existed. You'd like to see Einstein's brain?
Yeah.
And so we found that he has more glial cells per neuron than the average man.
So these are the nerve cells, and these are the nuclei of the glial cells.
Her hypothesis and measurement of the elevated number of glial cells in Einstein's brain fueled a controversy about the role of glial cells.
Brains are built of two kinds of cells, nerve cells and glial cells. And all these years we've thought that glial cells were just the support cells, the nurse cells, so to speak, for the nerve cell.
What Dr. Diamond had noticed in her research was that when rats are raised in enriched environments, their brains grew, but also showed increased numbers of glial cells.
I took that with the hypothesis that Einstein would have more glial cells per neuron than the average male, and we found he had significantly more. Caused a stir all over the world. In the first place, that it was a woman who found it.
The popular press was thrilled, but many scientists were highly critical of Dr. Diamond.
It goes on and on. Before it turned into what Diamond sometimes called a monstrous experience, it started as a simple case of curiosity. Findings were provocative. Einstein's brain.
Some criticized the quality of the 11 younger average human brains used for the comparative study. Others objected because apparently Einstein never gave permission to have his own brain preserved and studied. But mainly, the criticism was for making any suggestion about the function of the glial cell based on Einstein's brain alone.
It's not easy to go out on a limb in something as important as that, but science leads the way.
At a time where everybody, all the neuroscientists rush to study neurons, which are kind of the workhorses, or people thought of them as the workhorse in the brain. But Marion Diamond, her work focused on the glial cells that are always described as the support cells, kind of like the ugly stepsister of the brain cells. Well, now we realize that these glial cells, including the glial cells that Marion found in Einstein's brain, are doing more than support functions, including cognitive functions, roles in memory, roles in critical aspects of brain function. And so really the finding in Einstein was one of the first to herald the current renaissance in our study of glial cells and their importance. For example, now there are magazines just devoted to the glial cell, institutes that study glial cells. There are granting agencies that focus on glia. 20 years ago would be unheard of.
25 years after the Einstein study, here's footage of a living glial network communicating via transmissions of calcium, direct evidence that glia are active participants in brain function. Another paradigm-changing scientific breakthrough.
With Marion, the brain just wasn't this glob of neurons and myelin and glia. The brain was this wonderful, wonderful thing, and capable of so many things, including, as she would always, capable of love.
When I teach students, it's always a great pleasure to bring up some of the things that Marion has produced or things that she does. But the thing they're most interested in, and this of course is human nature, Not what she has done, what I've done, our relationship. When do we see each other? What do we do? How do we feel about it? Never underestimate the personal, the emotional, the limbic. Marion is my mate in every sense of the word, even though we are apart most of the time. We spend our lives 350 miles apart. Marion, of course, at UC Berkeley, and I'm a professor here at UCLA. And so we put up with this kind of interrupted relationship. And yet, over 30 plus years, it has worked for us. Marion, of course, is my second wife, my second marriage. At that time, Marion was married to a very fine man, a nuclear physicist, Dick Diamond. 4 children. The youngest had just left home. She was in the process of separating. It was 1979, and Marion's group, for some reason, they invited me up.
So, how did you meet Arnold Scheibe?
How did I meet him?
Yeah.
He came to give a lecture in my course.
I don't know what I expected, but I certainly found a very dynamic, blonde-haired young lady. She was truly the golden California girl who had a head on her shoulders.
He had lost his wife about 3 years previously.
I met Marion.
He had taken care of her for 27 years.
And it was a wonderful reawakening.
I was driving him up from the airport. Oh, everything's so beautiful. You know, he was like a bird coming out of a cage. He just loved the Bay Area. He loved everything, and that was really very different than most male scientists. They're not emotional.
And I apparently was a surprise to her because she expected somebody very formal in a bow tie.
Then he gave a beautiful talk on how the thalamic gates open to the cortex.
Well, I think that perhaps the most important thing that we have shared and continue to share is our love of the brain.
He knew his neuro, and he was kind, and he was fine.
And then she had some exercises to do. She had a class.
I told him, I'm sorry, but I have a Tai Chi lesson.
And she wondered if I'd wait in the library.
He says, I want to go to your Tai Chi lesson with you. Nobody had ever gone to my Tai Chi lesson with me.
I ended up on the floor in my suit coat doing exercises with them.
So you fell in love?
Yes.
We were married down here. As a matter of fact, right at the end of the pool. And as I recall, next morning early, I put Marion on a plane because she had to teach that day.
Anybody tell you that love is good for the brain?
Only Marion Diamond would use love in science.
Scientists don't study that because it sounds too squishy. But there's something there.
Marion began her research on love as a methodological solution to a very real scientific challenge.
We got our rats up to 600 days, equivalent to 60-year-old people. We put them in enriched environments and impoverished environments, and we showed changes. And I was invited to the German Academy of Sciences to report this, and one old German professor said, your California rats aren't very old. And I said, all right, we've got to see what we can do to keep our rats a little older than that and show that enrichment will affect them. What was missing, I felt, was what we call TLC, tender, loving care. So we decided to give loving care to our rats. And how did we do that? My technician and I would take a rat while their cages were being cleaned, and we'd hold them against our chest, and we'd pet them, and we'd talk to them. We dared, in scientific publications, to say love. Because when we had our rats got their love, they lived longer. We got them up to 900 days. But the point is, we could still show changes in the brain in rats that were equivalent to 90-year-old people.
At 900 days of age, we found distinct, significant differences between the enriched and the non-enriched. The plasticity of the brain was there, we found, actually at any age. Young, middle age, and extremely old age.
It is a message of hope. Here we have data, contemporary data, which suggests that the best may still be yet to come.
We always kidded why Arnie and I continue at this. Can we say our ages?
A couple of 900-day-old rats here.
It's that very basic, simple concept. Use it or lose it. If you don't use it, you lose these precious nerve cells that make up our brain. Getting that concept across to everybody helps them whether they're 5 years of age or close to 100 years of age. Would you like to put on your gloves so you can look at a brain? These are your gloves, those are your daddy's gloves, and these are my gloves.
I met Dr. Diamond in 1987 when I was a freshman at Berkeley, and she gave a 1-hour lecture when I was 18. And that talk inspired me to go into medicine, and now I'm a pathologist using what she taught me every day. But now I have a 4-year-old daughter. Indigo's not going to be able to take the classes that I took. My daughter is asking me questions like, is your heart beating all the time? So I wanted her to come and talk to the professor who inspired me.
I'm going to do this and take it out. And it's going to drip for a minute, so I'll try to not make too much of a mess. And you know what? Now your brain is looking at another brain. Do you feel it doing that? Does it know that it is?
You know, I think about my daughter and it gives me a new perspective on my admiration for what Dr. Diamond has gone through to get where she is, and hope that, you know, she can inspire, you know, a whole new generation of women scientists.
Now, how do you hear? This little part here is hearing. And if I take that out, no. So you see, that's the beauty of all of this part of the brain. It has different functions. Now, you're talking, so this part of your brain's working well. You're hearing me, right? And what's this doing? Is yours working well there? How do you know the back of your brain there?
Because I can see.
You can see. Good for you. Isn't that fun? To think all of that's going on in your head? This can be seeing, and this can be hearing, and this can be speaking. Oh, the satisfaction of learning. I look back now, and I remember when I was a first-year graduate student, studying brains, wanting to. It wasn't that common. In the medical school class, we had 5 girls out of 105 students.
In 1953, Marion Diamond was the first woman to earn a PhD in anatomy from the University of California, Berkeley.
Later, when we first started to write up my work, putting the titles at the top of the page, Kresh put his and put mine in parentheses, and I said, "David, what are you doing?" And he says, "But I've never written with a woman before. I didn't know what to do." I said, "Well, I did the work." And he changed immediately. I mean, he was really my friend all along. But we had a lot of education to do when you bring a woman into a male field.
There were other incidents of gender discrimination in Marion's career, but she didn't like to talk about them. What she would say instead was never learn bitterness, because you are the only one who suffers.
Hello, Dr. Marian. Hello, Marian. How are you? I am loud. Do you still recognize me?
We worked for years and years on how the environment affects the brain.
Hello to Dr. Marian.
We had established how the brain can change with good diets and with exercise and so forth. But we had only been working with rat brains, and I wanted to go to human beings. What I wanted to study were the most impoverished children I could think of in the world.
She called it the Enrichment in Action Project.
The monks in the local convent were feeding them once a day. They lived in the forest, they had no houses, they begged. Tragic.
She started to apply everything she knew about the rats and changing their brains with enriched environments, but applying it to people.
This was applied science, different from anything I'd done before, leaving the lab for the messiness of the real world. It wasn't research, it wasn't for publication in a journal. I wanted to see if our new understanding of the brain would really make a difference in people's lives, applying scientific knowledge to practical problems. That's what I wanted. So we've learned with our experiments that you have to feed the brain before it can do anything. So we said diet was number one.
Marian's Enrichment in Action program continued for over 10 years. She raised the money for it. She traveled back and forth to Cambodia repeatedly. She put to use the principles of enrichment from her research findings. This was science and philanthropy.
You should see them now, 10 years later.
Now I just finished the exam of university in year 2, and next month on start year 3.
5 of the students are finishing college, and more are on their way. A whole different life.
Yeah, wow.
And now we want this to be role models for the next generation.
Fantastic.
And that's what looks like is going to happen.
I'm just graduate from high school this month, and even the English before I don't know how to say hello, but now I can read and write.
Science to stimulate the brain, to improve your life. I just think it's magic the way things work out with cooking. But absolutely essential to measure things correctly. Women scientists make good cooks. Did you ever raise chickens? Back in La Crescenta, where I grew up, where we had chickens and goats and fruit trees, we always were barefoot and we always wore our little sunsuits because it was healthy for you. So there were 6 of us and we were all different exercises, and I was the youngest. You never know when you're going to get a new idea. When I was young, I went up and sat in the hills. I said, I'm just a little speck of sand in this big world. And what do I really want? When I saw my first brain, I saw that when I was about 15. One morning, my father asked if I wanted to go with him to the Los Angeles County Hospital. He was going to be visiting a patient. And so we were going down the corridor and we were passing rooms, but one room had the door a little ajar. And on the table inside the room was a human brain. Just the sight of that brain. And I said, those cells can create ideas. And that was embedded in my life. It still is, and we still don't know how, but it was enough to know that if I was going to study something, I wanted to study brains. It just blew my mind, the fact that a cell could create an idea.
Did that stay with you consciously, do you think, from then on?
Oh, very definitely. I mean, it still does, and we still don't know. And we don't know what questions to ask to find out. The marvelous things that this brain can do. The beauty of the brain, the art of the brain, as well as its science. And that's what I loved and still do. All right, I'm going to read my letter about my status with my field at the moment to my colleagues and friends. First and foremost, let me begin by expressing my sincerest appreciation to each and every one of you. I've had the privilege of sharing your...
Good evening, ladies and gentlemen, and dear friend. I was asked to speak because I've known Marion Diamond for 41 years.
Though I'd love to continue as before, I think I'd better pay attention to what my anatomy is telling me now. Thank you for coming, each one of you. Very, very fond memories attached to everyone. I want to be remembered as a vital, competent teacher who loves sharing with her students and providing useful information rather than one who is possibly slower and uncertain mentally and physically. You know how much I will miss you, each one of you. With affectionate best wishes, Marion. I'm not the lady I was 20 years ago, but I'm not going to hang on to the negatives. I never have. I would appreciate a professor that I had that recognized that we're changing and we're getting older.
In a sense, we're trying to understand what is this new time of life called age or extreme age. Mankind in general has not had the chance to experience it before. And Marion is, I think, very wise in wanting to nurture the image that she has represented for her students. She is not only neuroanatomy and anatomy, but the love of these topics. And I think they carry away that love And that's how she wants to be remembered, as a passionate, interacting individual. And I'm all for it.
Thank you.
Okay.
I do not understand why all people don't want to be teachers, at least part of a lifetime. I thoroughly enjoy talking to students of any age who want to learn and feel the light go on.
Marion has taught science to an estimated 60,000 students, enough to fill this stadium. Not even counting her YouTube audience, 60,000 in person over all her years. This is her legacy. Beyond any single discovery will be the continued discoveries from any of the people who studied with her and experienced the joy of curiosity.
Marion Diamond introduced me to a topic that has fascinated me for a whole career now, and probably for the rest of my life. And today, I have my own hat box, and I introduce the brain in that same way to my students. And it's like bringing her into my classroom. And that's the best way that I can honor her and all that she's given, not only to me, but to the thousands of students that have come through her classroom.
Don't you just love the brain?
What a wonderful woman. And as you may have understood now, the key behind glial cells is really the plasticity of the human brain. And here are like the glial cells that we know about. They have names that you may not have seen before. And I'll give you a short introduction of what they do in the brain. And it's very much a field that is in development to try to understand how cells can interact in the brain beyond neurons is really a future key to understand how the brain works and recover and change. So you have astrocytes, you have oligodendrocytes, you have Schwann cells, you have microglial cells, and you have the parenchyma. Because I like, to show how beautiful are those cells. I have those very nice histological sections where you can see that they have not only a name, but also a beautiful visual organization, where you can see here the microscopic picture of an astrocyte. And the first time people saw those cells, they were like, oh, well, it's probably just for physical support of the neurons, so they just don't fall and they get like their shape and their organization in the cortex. But further research really revealed that there is way more behind the contribution of astrocytes than just like physical support for the cells. For example, the astrocyte provides a nutrient to the neurons, such as the glucose that is necessary to function, or lactate, and they're able to modulate the blood flow. And you know that the blood flow is essential in the way we understand the signal derived from functional neuroimaging. And they can do also even more than that. They can modulate the synaptic exchange by uptaking or releasing glutamate, ATP, and GABA. And through this modulation at 3 levels, where they can feed or starve a neuron, modulate the blood flows, that will also feed the neuron, but really change the communication between neurons by uptaking or releasing glutamate, ATP, or GABA. They modify the very functioning of the human brain. And by doing so, they open the question on is really the neuron the center of the functioning of the human brain, or is this work of the functioning of the human brain shared, like, the first place on the podium? Would it be shared between neuronal and glial cells? Well, this is an open question that I'll let you think about. So for a long time, really, we were really interested about all the biological mechanisms that are related to the neuron. And we had sort of like just, half of the picture. And you can see here, how the blood interacts with the neuron, brings things to the neuron so that it can work out its action potential. However, more recent work, thanks to really the enlightening of the research that was brought by the research of Marian Diamond, push people to investigate more the role of glial cells. And as you can see, the biological and chemical and the chemistry of those cells is of equal complexity. And it's really an interaction between neuron and glial cells that is key to the functioning of the human brain. Now, advanced investigation has been able to dissociate the activity between neuron and astrocytes. And the only way to do that was to find a method that will isolate the activity, like the stimulation of a neuron, from the activity of the astrocyte, and isolate the activity of an astrocyte from the activity of a neuron by stimulating an astrocyte without stimulating the neuron. And the only way we've been able to do that is through a method that came out like quite recently, which is optogenetic.
In optogenetics, what we're doing is we're putting molecules that convert light into electricity into neurons, the cells of the brain. Then when we shine light on those neurons, light gets converted into electricity and allows us to turn on or off those cells. The goal here is to find a way to control the electrical activity in some cells and not others in the network. To do that, we had to turn to the natural world. It turns out that throughout all the kingdoms of life, in plants and funguses and bacteria and so on, you can find photosynthetic or photosensory molecules that convert light into electricity. So we borrowed these molecules from nature, and then using tricks from the field of gene therapy, we can put them into neurons. Now, these molecules can convert light to electricity, and they do it just in the neurons that we want to control, and not all their neighbors. So we can deliver these molecules to some cells and not others, and then we shine light on the network, we can turn on or off that subset of the cells. If we can turn on or off a set of cells that's embedded within this dense matrix, we can figure out how do they contribute to a behavior. For example, if we can turn on a set of cells, we can figure out what kinds of behaviors can it initiate. If we can turn off a set of cells, then we can delete it momentarily and figure out what it's necessary for. So by being able to dial in information into cells in the brain and to delete them, we can try to figure out how they contribute to networks and the behaviors and diseases that arise from brain computations. We can hunt down the exact set of cells that are contributing to a specific disease state, or which, when activated or shut down, will remedy that disease state. That's really important because right now a lot of drugs are developed that target molecules, but molecules are found throughout the brain, and in fact many cells in the brain might be very molecularly similar to one another. If we can target circuits in the brain, though, we might be able to develop much more specific drugs. Imagine if we could hunt down the exact set of cells in the brain that, when activated, remedy a brain disorder, and then we can go in and look at the exact molecules in those cells. Maybe we can find drug targets that are much more specific than existing ones. You might also imagine that we could use optogenetics to directly control brain circuits in patients with brain disorders. Electricity is used to stimulate the brain in deep brain stimulation. If instead we could actually aim light at certain cells and turn them on or off, we might be much more specific. Rather than using electricity to turn on and off the cells of the brain and have many kinds of cells activated, the ones we want to affect as well as all their neighbors, if we could make just one disease-associated subset associated with light, and we can turn them on or off, we might be able to treat them with much more specificity. So far, optogenetics has had a lot of impact in the scientific world, but it hasn't been used in any human patients yet. There are a couple reasons why. One is that it requires a gene therapy to deliver the gene that encodes for these light-activated molecules into the body. Currently in the US, there are no FDA-approved gene therapies. In Europe, there's just one. Another issue is that these molecules come from organisms like algae and bacteria. And so if we are putting these molecules into the body, would they be detected as foreign agents and attacked by the immune system, for example? What we need is a paradigm shift in how we think about treating brain disorders. And one of our major stances is that we need new technologies if we really want to either understand the principles of treating brain disorders, hunting down the exact cells in the brain that can help us treat brain disorders, or to develop new modalities, new forms of energy, new strategies for treating brain disorders by correcting the computations within the brain.
Impressive. And that's exactly what they did here. They used optogenetic to dissociate the stimulation of neurons from the stimulation of astrocytes. And they measured functional neuroimaging, which usually we associate to the activity of neurons. And so what happened really, as you can see in B, D, F, and H, is the stimulation is characterized by the blue line, and the end of the stimulation is indicated by the yellow vertical line, okay? And what you can see is like, if you stimulate a neuron in B, you are going to have an increase of the fMRI signal right away until you stop stimulating, which is a yellow vertical line, and then like the functional MRI signal decreases. In D, what you have is the stimulation with light of the astrocyte. And you can see that when you stimulate the astrocyte, you also have this increase of the functional MRI signal, very significant. That stops when you stop the stimulation of the astrocyte. Now, if you look at the slice in the brain in A and C, you can see that the activation colocalized, but are not exactly the same. So next time you look at fMRI, just ask yourself whether this is really activation of the neuron or activation of the astrocyte you're looking at. And if this is the activation of the astrocyte you're looking at, and this is related to something your subject is doing in the fMRI, is, the fundamental cell behind the function of the astrocyte then? And I find this a fascinating question. Similarly, you can do the same thing with deactivation or inactivation, whereby you can see here that stimulation can also depolarize, like can also deactivate a region and reduce the signal, the fMRI signal. And the same goes for neuronal stimulation and astrocyte stimulation. And you have the link on the right for the video I just showed to you. Next are the oligodendrocyte. So if the neuron was like kind of like, you know, the ideal structure for localizationist people, associationist and holistic researcher, those who believe in that theory, will be much more interested in connections. Now, white matter connections, in order to optimize their speed, really need to be myelinated. And that leads to a change in the way the conduction is happening in axons, and you have what we call saltatory conduction that is way faster than unmyelinated connection. This is really a wonder of brain evolution. So, and this was necessary because brains became bigger and you have larger distance to cover. As a counterexample, for example, if you take the fly got a very tiny brain, no need to myelinate the connections of the fly because the distance to go from by the action potential is actually much smaller, so the connection of the fly are not myelinated. But the bigger brain like us or the primates or the mammals need myelination in order to cover the large distance between neural cells. And so in order to do so, they needed like a cell that produces myelin and cover up the axons. And the main star here is the oligodendrocyte that can extend multiple branch to contact the nerve cells axons and wrap them with myelin insulation. Okay, so that like, if you see the connection here in purple in the picture, the oligodendrocyte here will be in green, and you see how it wraps up around the cells, or a bit like the net of Spider-Man, if you want. And this is important because we know that change in myelination can lead to change in connectivity, and change in connectivity can lead to a change in behavior or learning new skills. So that's really also a fundamental cell behind plasticity. And so this oligodendrocyte can connect up to 50 axons, and they're going to wrap, as I said, the axons with a myelin sheath. And the wrapping of myelin action is complemented by Schwann cells, which are other glial cells that do the same job, but for a single axon. I thought like, you know, you'll be enjoying also to have like an histological section where you can see here a section cutting through an axon, and you can see really in dark gray the myelin sheath around the axon that is really like wrapping it and insulating it really. Another cell, another like glial cells that also you need to know about, and that is probably the most fascinating one, is microglia. And so the microglia you can see here in red, right next to an astrocyte in green and another neuron in green, is much smaller than an astrocyte, and that's why it's called microglia. And those microglia travel independently. They can go from one part of the brain to another part of the brain. They're not attached to any structure like oligodendrocyte, Schwann cell, or astrocyte. They really move the way they want. And they constantly circling a territory with extended arms, repeatedly tapping all axons, dendrites, and synapses, looking to detect any suboptimal functioning. It is the most active cell in the brain, and that's why it captures the attention of scientists. And you can see here on the top right the kind of job they're doing. They contribute to the immunity of the cells of the brain by detecting like an external agent. But they will also work on the synaptic and the neural pruning, which we know is really the key to brain change and plasticity that make you who you are and not somebody else. And to do so, they're able to literally change their shape according to whether they're doing a synaptic pruning, a neural pruning, or doing the immunity through a technique that we call the polymorphism. At the bottom right, you have another picture of the microglia that you can see here. They're in dark blue right next to other cells. Last star I would like to talk about is ependyma. So the ependyma is usually found in between the white matter and the ventricles that are indicated in the brain that you have on the right of this slide. So the cortex is in red, the white matter is in white, and you see those dark bubbles inside the brain. Those are the ventricles, and they're full of cerebrospinal fluid. So the ependyma is actually producing this cerebrospinal fluid, or CSF in short. And if you zoom in very close, you'll see what you see on the left part of this slide is like they have a cilia that will move and help with the flow of the cerebrospinal fluid in the brain. And that's the job that they have, is to help this flow, and the cerebrospinal fluid allow to clean up a little bit the brain. Interestingly, it's been demonstrated that after a stroke in the brain, you will definitely lose neurons and part of your brain will be damaged. Those cells can convert into neuron to support the functioning and the recovery of the human brain. How fascinating is that? By doing so, demonstrating again that glial cells are really a crucial key to the plasticity and the change of the brain, making you who you are. So this is it for the glial cells. Next lesson that we will have will be on the epistemology, where we're going to learn that what we know is fundamentally biased by our ability to observe it, and what we cannot see is clearly much bigger than what we can. I just take this opportunity to remind you that it will be the 12th and the 13th of November, actually. We'll have a brain structure and function special issue symposium on the structural connectivity of the cerebral cortex. I know you won't be available during the live event, but you can actually assist this event in replay on the CNS seminar YouTube channel. And that every Monday morning from 9:30 to 10:30 a.m. we have the Neurochino. Grab your coffee and eventually an interesting paper that you saw last week, or just sit down and learn about what happened and what's exciting in science last week. And this is every Monday, and you're very welcome to connect. All the links to connect can be found on the website of clinicalneuroanatomyseminars.com. As a reminder, there will be an evaluation and grades for this module. My evaluation, the grade I'll give to you for this module will be average with Principle of Neuroscience Part B from Professor Valesi. The way I evaluate your performance is a mix between your attendance to the lesson, your participation, and a little project. Your attendance being like mostly the practical, so I can see you were connected. Your participation being how many questions you can ask. You can ask questions directly on the YouTube channel, and I will reply to them. I already saw a few questions, and thank you for that. But you can also ask those questions on the Neurostar forum that I open. I open a channel on the Neurostar forum for you. And the last part of the evaluation is a little project. It is an essay that I'll ask you to write and send to me before the 11th of December on the subject of brain hierarchy. So I need you to choose a hierarchy of your choice in the brain, such as a visual ventral stream, anterior-posterior organization of the frontal lobe, the temporal semantic system. You have a lot of hierarchy in the brain. Please surprise me and find some things that I don't know. What is important is that you write 1,500 to 2,500 words about it, not including the reference section, but still including references. I'll need you to write these 1,500 to 2,500 words in 5 different sections, no matter the size. Starting with a general introduction where you will get me excited about the hierarchy in the brain of your choice. Then you will tell me a little bit about the anatomy of this hierarchy. Then I want you to do some research to see whether this hierarchy is lateralized, and if you don't find anything, suggest ideas on how you would investigate the lateralization, the brain lateralization of this hierarchy. Next, I'll need you to make a little section about the link between this hierarchy and brain evolution, and finish with a 5th section, which will be a synthesis where you show me how your general introduction, the anatomy, lateralization, and evolution of this hierarchy are working together, and open up the subject for future research. Make it simple, interesting. Your parents must be interested about it, and I wanna, that's really one of the key behind good writing, is probably making it interesting. You send it to me to my personal address at michel.thiebaut@gmail.com. And with this, thank you for your attention, and look forward to see you during the practical